Hi there, I'm graduate teaching assistant Aldo San Pedro, and I will be walking through a tricky 3-point test cross. This is problem number 15 from Chapter 5 in Genetic Analysis, 3rd edition. This question demonstrates that a 3-point test cross can be tricky. Theoretical knowledge of linked genes and independent assortment are required. Before we jump in, I will emphasize that I will approach this problem as if I am solving a typical 3-point test cross. On that note, I will walk through each question, A through E, once we obtain all of our required information. Also, I will review repeated steps quickly, so I encourage pausing the video and taking notes at an appropriate pace. My first objective is to duplicate the table to the left and simplify the phenotypes into characters that make solving this problem a bit easier. Here, I transfer the progeny numbers and total progeny. Next, I replace phenotype traits with their corresponding character as described in the question. Tunicate can be labeled T. Do note that for some 3-point test crosses, you may have to assign your own characters. I replace glossy with capital G and the trait non-glossy with lowercase g. The same technique follows for legold and legalless. Capital L and lowercase l are used respectively. Next, our goal is to describe each kernel's phenotype pairs as recombinant or parental. This will aid us in calculating recombination frequency and building a genetic map. Genetic interval pairs for this question are T through G, G through L, and T through L. Our parental phenotypes include homozygous dominant T, G, L, and homozygous recessive T, G, and L. I assign whether a pairwise trait interval is parental or recombinant based on if the phenotype pairs match the parental phenotype pairs. For instance, capital T and G are parental, as are lowercase t and g, so I assign these pairs with a P for parental in the column to the right. Then, I label any phenotype interval deviating from either parental phenotype pairs with an R for recombinant. I repeat these steps for the next pairwise intervals, G through L and T through L. Then, I begin adding recombinant progeny from each pairwise interval. For column T through G, I will sum progeny totals of 18, 20, 22, and 23. These numbers sum to 83. I repeat this step for the remaining 2 columns. My results are a sum of 246 for interval G through L and 247 for interval T through L. I now have the information required for calculating approximate recombination frequencies for each pairwise interval. Do note that we have not been able to identify any possible double crossover DCO events yet, so we are unable to include them in calculating our largest interval. We'll get to this later. To calculate our recombinant frequency, RF, we multiply 100 by the quotient of our recombinant gametes over total progeny. The expression will also describe map units, MU, or centimorgans, CM, for each interval. I use the formula for each interval and obtain the recombination frequency percentage for each pair. Something interesting is happening here. Recall that unlinked, independently assorting alleles have a recombination frequency of 50%. These 2 recombination frequencies are suspect. Recall that unlinked, independently assorting alleles have a recombination frequency close to 50%. This relates to Mendel's second law of independent assortment. Even if we identified and included double crossover progeny for T through L, our recombination frequency would be 65.8%. I include my calculations with the anticipated double crossovers. The locus for allele L can be on another chromosome, or distant enough from the other loci on the same chromosome for independent assortment to occur. If we were to map this scenario with 2 chromosomes, it would look like this. And for the other scenario, where all loci are on the same chromosome, Do note that theoretical recombination frequency cannot be greater than 50%. However, genetic distance can be more than 50. If genetic distance is really greater than 50, the distance cannot be determined by recombination frequency alone. You will need additional loci to determine. This is why a loci L can be on the same or different chromosome until further testing. Let's tackle some questions now. Question A asks us if there is evidence of genetic linkage among any of these gene pairs. I encourage you to pause the video and attempt to solve the questions on your own. T through G are linked. T through G recombination frequency is below 50%. It's actually 16.6%. Among the least frequent progeny, recombinant T through G are present, and not present in the most frequent progeny. Question B asks us if there is evidence of independent assortment among any of these gene pairs. Yes, L is unlinked and assorts independently among the T, G gene pairs. Among the four most frequent progeny groups, capital T through G and lowercase t through G remain parental, whereas L recombines with no significant change in progeny. Also, the recombination frequency is very close to 50% for intervals containing gene L. Question C asks us to write the genotypes of F1 and F2 plants. Recall that alleles for linked genes are written on opposite sides of a slash to indicate the linkage phase. Question D asks us to calculate the recombination frequency for our linked genes T through G. We already did this step earlier, but do note that the question is not asking for the percentage, so answer with a decimal. Our recombination frequency for T through G is 0.166. Our last question, question E, asks us if all three genes can be located on the same chromosome. Recall that we solved this earlier. Yes, all three genes can be carried on the same chromosome. T through G are linked and are on the same chromosome. Meanwhile, gene L may or may not be on the same chromosome. If it is, gene L is separated by loci T and G by a distance greater than 50 centimorgans or 50 map units. Thanks for watching, and I hope this video helped illustrate the nuances of 3-point test crossovers.